Hello, welcome to the second presentation of our course, Medical Genetic. Let you know that our course is mainly talking about DNA variation and the impacts of the DNA variation to our health. I mean, it's the main thing talking with the disease and the healthy different. And before we go deeper in talking about the mutation and impact, so in this presentation, I want to remind you about something basic of the cell biology. And from here, I want to make you have the better sensation to where is a mutation come from and what impact, how strong the impact to our health. So let you know is the cells is the most complex thing in the world. No, I think it's the most complex thing in the universe. And what is the complex is the most easy to break. So they say that I'm not trying to recall all the thing in the cell biology because it's quite very big. So I just focus on DNA and something related with DNA and something like discuss about the central dogma and recall you about that thing. So before we go deeper about the cells, I want to come back to the last presentation about the definition of the medical-genetic. So I try to have another view of the medical-genetic. So in here I showed a picture. For example, I have one disease, and that is specific disease, and not related with infectious or something. And you see that is disease and the case they have a small fraction in the population compared with the healthy people, the blue one. So because I'm kind of worried about my health, that my health is the most important in my life, I think. So I want to explore what is different between me and the healthy people. And I check and I see that some people is similar with me. Like I working at night, late night, I drink a lot of beer and not do exercise. So I mean that the environment between me and some people in the healthy group, they are same. So is the phenotype or disease outcome is a sum of so we can explain by the sum of the effect from the environment and gene. So in here is environment is totally same, for example. So we can explain by the gene. So my question is, go to the next question is, what gene? What gene make it different? What gene make it different? So that is the problem with me. Because you see that in our cells, we have the 1/2 of the genome haploid. We have 3 billion base pair. 3 billion base pair. And another question is, that kind of disease is caused by the single or multiple gene. And in my cells is not only a single haploid. We have another haploid, that means diploid. Haploid from a set of the chromosome from mom and a set of chromosome from dad. And how does the effect sooner that allele effect? So it's quite complicated. And another question very important in my life, if I have disease, is what happened with my next generation. So if I have the boy or the girl, is which one will have effect with the disease? Or if I have positive birth, so can my child avoid the disease or not? So is we can, right? So we can use the IVF technology and using the PGD, I mean, so we had take some cells and check if that cell is normal or not, and we transfer to the to my wife. And maybe in some time you will have some facts. You will match some these facts. Like, what if your wife or your husband have genetic illness, brother or sister? So that means your partner also have some bad gene in her or his DNA. Or in some case, or his or her father is affected by Agent Orange, the mutant chemical in the Vietnamese war. So that means his or her also have that bad gene. So if you are not the case, you're not worried. But if you are in the case, you very so much worry about this situation. And the final question is, I wonder, all of the thing about is, can you give the advice after this class or not? You have to develop the knowledge and also the sense by combine all the stuff you learn from genetics, from the cells, from the biochemical, biochemistry, to explain and give the advice for the case you maybe you counselor, I think. Okay, now we go a little bit briefly about the cells. So here you see is the most complex thing in the universe, I think. But I'm not trying to cover all things of the cells in here. So we just focus on the DNA I wanted. So you see, it's the most of DNA is stored in the nucleus, and all of the protein, all of the gene is in the DNA in the nucleus. But it's not only nucleus. We have some small fraction of the DNA in the mitochondria here. So mitochondria also has some plasmid. Is about the size of the plasmid, about 16 kb, and is coding for some gene is necessary for mitochondria function. But you know the function of mitochondria, right? It's a powerhouse of our cells. They provide the energy for the cell can work, and also whole body also relies on the ATP, the power of the mitochondria. So if we have the mutation in that mitochondria DNA, so what you think of we have the effect? That's a huge thing. We have not enough energy for the function. The most effective thing will be the nerve system, the brain, because the brain is the most used energy organ in our body. So you see it later. Okay. This is a very familiar with you, right? So gene is a basic unit of inheritance. So we transfer to the next generation, and they contain in the chromosomes and consist of the DNA from the simple form, like the string like in the top one, and they more condensed in the chromosome form by using the histone the histone protein here. So humans, after in thousand or 1001 year in the U.S., we had a very big project finished genome sequencing, human genome sequencing. So after that, the scientists, after analysis, we can see that human have about 20 to 25,000 genes, but that number is not very. I mean, it's still a lot of debate for that kind of number. And the next slide will show you that kind of thing. So if you ask how many genes human have it, so the number you will see is about 20 to 25,000 genes, but kind of small with a 3 billion base pair. But it's how is the scientists discover But recently, another scientist tried to return that question, and they found that number of genes may be changed by different definition. For example, in here we have a protein coding gene. Protein coding gene in here, and we have the column is shown by a different database. In here we have the gene code and ensemble receipt from the NCBI, and this is the new one, the chest thing. And the protein coding gene, we have about 20,000 around that. But we have another thing, is some kind of the gene, but they don't make the protein. They make the RNA for regulation function. And also we have the pseudogene right here. The pseudogene is some fake gene. That means some gene, but they die already in the evolution process. And the total transcription, I mean, is the number of the transcript. I mean, from the DNA to the RNA already. But we still not sure about the function yet. Still a lot. So by the different definition, we can have a different number of the gene. So a lot of the gene, you see this number, a lot of the gene is involved to the regulation function in the cells. And this is the structure of our DNA, the double strand, double helix here. We have the this is a one double strand DNA, and this is the structure of our DNA. We have thymine, adenine, cytosine, and guanine, just 4 base, and the coding for a very complex system in our cells. And this is the structure of DNA. The structure of the nucleotide. We have the postbase, we have the sugar, and we have the base. And I want to mention one thing. For example, in here we have, for example, this is A. This is maybe this is a same strand, and this is an antisense. Remember, that's right. And this is A, and it should be, this is a T. And this double strand is come from mom, and we have another come from our daddy. And our daddy also have the same thing. But if I want to denote that my DNA is a mutation is A and T, for example, A and T. A is a come from this. And T come from where? Is not come from here. T is a come from this strand. This one, T. And this should be in the antisense of another double strand is a A. So a lot of students still misunderstood about this stuff. People just AT is this AT. This should be A, this and this A, and this T. Okay. And this also is very basic in the cells. Like we have DNA replication fork. So we have the leading strand and lagging strand. So continuous and discontinuous in a DNA replication. Yeah. And the DNA replication, we have a lot of the replication bubble. I mean, it's replication not only happen in one place in the strand of DNA. They occur in the multiple sites, a lot of sites in the chromosome to speed up the DNA replication. And this is a central dogma. So from gene to the protein. So in here is chromosome in the top, and we can see a DNA should be the eukaryote, so we have the exon and introns form. And in the transcription, we have the primary mRNAs, and after splicing, we have the mature mRNA, and they go out of the nucleus by nucleus 4 and go to ER to the ribosome to make a protein. So you see, this is quite simple and all you know, but you know it. Nowadays, the scientists still discover a lot of process a lot of process going parallel with this thing, and that will make you a lot of the new knowledge about the cells. For example, like it's how we have just 25,000 gene, 25,000 gene, 25,000 gene, for example, with the old definition. With each gene, we have a different promoter and different ATG or something. But in some case, we had a mutation. Some part is don't have gene at all, but they also have the coding reason, but they don't have the promoter. But if a mutation happen, that gene will be wake up. A lot of gene will be wake up, and that's normally happen in the cells. So they will increase the number of transcript in our cell, but that transcript will be, I don't know, is they some of them they will make the protein. They will make a protein, and that protein is have no function. Or if they have function, they are wrong function and make the our cell is got the disease. Another thing is splicing. Splicing in the process. Splicing in the process, we remove intron from the exons to connect the exons together only to make the mature mRNA. So if the something mutation happen, for example, we had some mutation in this point, so that some enzyme cannot recognize intron and exon anymore. So this intron will be retained and move to the next, still keep into the mRNA. And you know it, in the intron we have a lot of stop codon, and the DNA cannot, protein it cannot express well. So we lost that protein, and if we lost the protein, our cell function also will be affected. For example, and like I just read this one several days ago. For example, when even we had a mature mRNA, the mutation also happened. So the adenine will be transferred to the inosine, and then they make the mutation in the DNA because of it's A will be by with G, but inosine can bind with a lot with the C or T, or back to the A. I don't remember exactly, but inosine can bind with several another nucleotide. If you know remember well about the biochemistry, you will remember inosine is a precursor of the adenine and guanine. So if we want to synthesize adenine and guanine, you need to have the inosine first, the one type of nucleotide in our cells. So this time talk about the transcription. So we synthesize the RNA from the DNA. Talking about the protein coding gene. So we have this in different cell type. We have the lung cell, we have the fibroblast cells, we have the nerve cells. So each cell have a different set of gene can be expressed. That means can be transcribed to the mRNA and make the protein. Only a small fraction of gene is actually transcribed. I mean, it's that gene, those gene is kind of very important, like beta actin. They make the skeleton of the cells. Any cell needs a skeleton, so actin should be expressed in any cells. So we call that cell is housekeeping gene. Yeah, and other gene may be specific for specific cells, and specific set of genes should be followed. And if the set of gene change, the, those specific cell will be no function. And this figure, this figure is show that the regulation system in the eukaryote, especially in the eukaryote, is very complex. So in here is um, we see the effects of enhancer in here, and you have a suppressor or silencer. So like the name says, enhancer is try to enhance the transcription of the gene. So if you have more mRNA, so more protein will be made for the cells. So the the special thing about enhancer is they localize quite up near or far from the promoter region of the gene. So they try to recruit some transcription factor. It's very complex system in here. So complex that this factor will be induce the RNA polymerase to process the transcription. Um, this slide talking about the DNA binding motif. You know, it's DNA bind with DNA that's by the complementary or RNA can bind to the DNA too. In the cell, we have the RNA primer bind with the DNA to prime for the DNA replication. But uh, there also very basic thing is the protein also bind very well with the DNA with the DNA. By the here we have the helix loop helix protein will bind with the group thing of the DNA here. But that happens only when the DNA in the form of the decondensation. That we know is that's euchromatin. So in the euchromatin in chromosome, they will increase the gene expression because of they quite open and the transcription factor will become to enhance their transcription. And in the opposite with the euchromatin, we have heterochromatin. Heterochromatin is in that a portion of the uh, in that portion of the chromosome, the DNA will not condense. They did not decondense. They condense. They condense. So in the condensed form, there's some protein or transcription factor that cannot bind well to the DNA and no transcription at all. And right now, the new information from the from the research, they show that different cell type will have the different sex. I mean, different form of the different reason of the distribution of the euchromatin or heterochromatin is quite different cells and different distribution of the open or closed in the gene. That also show that in the different cells, in the different specific cells. And the closed or open chromosome DNA. So there was, in fact, by one factor that we cracked around already, that is based on the acetylation of histone. You know, it's DNA bind well with the histone. And if the tail of the histone molecules have the one chemical group is the acetine. If the acetine bind to the histone, if the acetine bind to histone and histone cannot bind with the DNA anymore. And the DNA, if not bind with histone, they will open. And if they bind well with histone, they will close. Because histone help the DNA is the supercoil. Uh, this one I a little bit explained already. This the splicing, but like we know that our cell we have the 25,000. I just use this number. So that's quite not enough. Not enough for the complex function of the cells, because any kind of activity of the cell we need a protein. Without protein we cannot do anything, because of so that 25,000 scientists think is not enough, and the cell is quite very smart. They have another phenomenon to help the cell can handle that kind of thing. So that's what we call this alternative splicing. In here, only one gene, we can make some kind of different mRNA, different form of mRNA by combine different set of exons in the same gene together. So in here is we have the 3 exons, and in here in the end we have mRNA with the complete 3 exons. But sometimes this exon will be removed because of this one is they not recognize, and they cut this one and they cut this one, and this exon 2 will be removed out of the mature mRNA. So we have a new gene. We have a new gene. We have a new protein with a new function. That's true. Like if you search in internet for each gene, you will see that each gene will have a different express mRNA, and we call it isoform. Okay. So that makes this our transcriptome is more diverse, and more diverse we have a diverse protein, and they help us to make the cells can conduct the very complex work in the cells. And this slide is genetic coding. So genetic code, you see that we have just 20. Actually, we have 21 amino acids, not the 20. But we have the 64. We have the 64 codon. That means 3 nucleotides make together to coding for one amino acid, and so that some different triple codon will be coding for same amino acid. Like for example, in the case of leucine here, we have a 4 triple codon coding for leucine here. But there's one thing I want to share with you. That is, for example, different species, especially for relationship like bacteria and human. So human maybe they favor for some codon only, and the bacteria favors for remain codon. Why? Because of the tRNA. So tRNA of the human and tRNA in the bacteria are kind of different. So they recognize specific the triple codon and it work more effective in the family triple codon. So if you want to express the protein in the E. coli, if you want to express human protein in E. coli, you have to transfer from the human, for example, in here to this coding. If you try to do cloning of gene. And next is translation. As you know, is a ribosome to like the machinery to conduct the translation to connect the amino acids to the monomer to polymer like protein. And after making the protein, after making the polypeptide needs to be going to the truncate. A lot of post-translation modification in the ER and also in the Golgi system. They will truncate the protein, they cut the protein, they remove some part you don't need it, or they can add something like they add some side chain by carbohydrate or lipid, or they can combine between the different proteins together to make their multi-subunit protein. And this is one example and the single mutation and effect to whole body. So that should be very important gene. Only one single gene, single mutation can make a whole body effect. So in here is related with collagen. Collagen is the most common protein in the cells, in all the body too. So in here we have the kind of the one a single strand of the collagen in here, one molecule of collagen. So they need to reassembling assembly to make the collagen fibrin, but the mutation make the glycine to another type of the amino acids. So the assembly will be not happen. So or happen but not very well. So here you can see the bone of the baby is the effect. Is not they kind of fracture or something, fracture or it cannot connect well because the bone is a bone cells is made from uh, connect connection tissue. This is made by collagen. So only single mutation can make a very large effect in here. And next, I want to remind you about the structure of gene and genome. Structure of gene is kind of simple, right? This is structure of the gene in the eukaryote because we have the intron here. And we have the regulations part like a promoter, and we have exon for the copying of protein, and we have a poly-A reason for the stop of the transcription. If the transcription comes to the poly-A signal, they will stop here. And we have enhancer in here. So actually enhancer is not necessary to be next to the promoter. They can very far from the promoter. Or new findings show that the enhancer can be localized into the intron also. The bottom figures show that about the structure of genome. So in our genome, maybe next slide will be so in here we have the single copy DNA about 45%. Single copy DNA is what? Is the DNA reason that you cannot find any copy in the whole genome. I mean, we have just only one sequence like that. But we have a 55% remain is a repeat one. That means the DNA sequence we can have another copy throughout the genome. In the large side, we have the disparate repetitive DNA, or small side, we have a satellite DNA, about 10%. So it's more better. So actually in here we have just 1 or 2%. It's very small. But remember, in intron we have quite a very big size, and we have an intergenic single copy. That's the part they are between the gene A and gene B, but the gap between gene A and gene B, that is intergenic part instead. And the large part is a repeat sequence. repeat sequence, we have the large scale like long intersphere element, or a short one, and another kind of different kind of repeat smaller. So I will introduce to you later about the dynamic of our genome by the repeat sequence. Next is we talk about a cell cycle. So in here is the mitosis, very basic, right? So we have the gap one G1. So in here we have the RNA and the protein expression, and the synthesis stage interface. We have DNA replication, and in the gap 2 we have a very important thing. Is we do reparation for we prepare for the DNA. If the DNA copy have some damage, so we have to repair protein come to fix that kind of wrong copy. And this process quite a different cell type. For example, intestine and lung cells. Because intestine is inside of our gut, they put acids and they very easy to break. So intestine needs to be very speedy propagation of the cell. And the lung cells, the same thing. The time for lung cell intestine is very fast, about just 10 hours from one cell can have two cells. But in case of the liver, so it takes a year to make the one cell can populate can make double cells. So this is state of mitosis. So in the final, we will have the daughter cell with a similar copy of the genome. So, but actually, it's not really similar. After each time of the doubling, the cell will have some nucleotide different with each other. Maybe 5 or 8 nucleotide will be different. So that is a mutation. Mutation will accumulate from this and to the next generation. And this is a state of meiosis. This picture is come from women, you see, right? Because of this is X, it's a big X, and this is quite small. This is figure show that. So in the X, the only one end is become the mature X to ready for the waiting for the sperm to make the zygote. So and also a very famous thing is a crossover between the sister chromatic in the meiosis that happen in oxide and also in the sperm. But we have quite a difference between the sperm and the egg. So you see that the old man can have the baby easily with the young women, but in the reverse case, the old women it's hard to get a baby right because of the sperm is they can make it from the stem cell, and the sperm cell is the only the new cell come from the sperm cell the stem cell in the man, but in the women the eggs after when the girl is the baby, the eggs is already made already. So that means the eggs is already old when the girl mature. So the eggs of the old women is kind of quality is not kind of good. So that you cannot use the eggs for fertilization. So that is quite a basic difference between men and women in the reproductive. Okay, so we finished this thing, and my question I can rewrite the report from you in the next before next presentation is the question: why the disease forms are rare, the uncommon thing in the population, compared with the healthy one. Healthy is more common compared with rare, compared with disease form. So please submit it on Blackboard discussion part. Thank you.